It makes your heart beat faster. All right, so how are you guys need to study that stuff, okay? That's not a wait till the last minute stuff. So have you made your little, okay, is that helping?
Is that the only thing we should study though, or should we still go through the chapter outline?
That's pretty much all it is. You know, it basically talks about each of the different glands and what it does and and that's just a way to narrow everything down. So that and the difference between water soluble, lipid soluble, and then how to make a thyroid hormone. Pretty much. Right?
Glad now that we're done with the HESI.
Yeah. Ugh.
Yeah.
Everybody always says that. Ugh. So, any questions on that before we go on to blood? We're good? This is one of my favorite chapters. So I get a little overexcited about blood because in my past life, I was a lab technician. So I started out my college career in community college. I got my associate's degree in medical lab. And that's how I put myself through the rest of college, working in the clinical lab in clinical chemistry and then hematology, and kind of a generalist person.
So you were a nerd?
No. I'm a nerd? Yeah, I'm a wicked nerd.
Are you a nerd?
Oh, yeah. Yeah. Science geek. I started as a phlebotomist, believe it or not, at 16 years old. Yeah. Back in the old days, they used to train you in the lab. So on the job training, I used to work as a secretary on weekends, checking in patients for blood tests in the lab. I thought, oh, this is kind of like this stuff. So I said, geez, will you teach me how to? So they taught me how to draw blood. I used to do cardiograms and urinalysis. So when I was 16 years old, that's what I was doing. That's how I put myself through community college. So yeah. I've been a geek for a long time. So the clinical lab was a great place. I really enjoyed that. And that's, I don't think they let you do those things anymore. But why? I was awesome. I was the one that they used to send when nobody else could get blood. I was good at it. So, yeah. Don't worry about age. Right? So, blood, I've kind of been close to it for many, many years. So what is it? Why do we have a whole chapter devoted to blood in an anatomy and physiology book? It's our transport system. It's delivery. Not only delivery of things that we need, but getting rid of things we don't need and delivered to the systems that are going to take care of that for us. So it's connective tissue. It's a liquid connective tissue. 55% of blood is mostly what? Plasma. Most of it is actually water. So 55%, we're going to talk about plasma in a minute. Plasma is not just water. Blood is thicker than water. Do you know why? Bunch of different proteins that are floating around in there, plus we have some cells. The other 45% of blood is mostly, believe it or not, red blood cells, erythrocytes. We call that our packed cell volume. And then about 1%-ish, it's a little under that, is white blood cells. So not only is blood for transporting substances from one place to another, but it's also a place where these guys hitch a ride. We already talked, when we talked about hormones last chapter, we talked about monocytes, right? What's a monocyte? We're going to talk about them more in this chapter. It's a type of white blood cell. There's 5 different types of white blood cells that hitch a ride in the circulatory system. So they go from one place to another via the circulatory system. They can go places and be told it's time to break down bone matrix, leave the circulatory system and do their thing. We're going to meet the other leukocytes, and their jobs are basically a whole bunch of different jobs doing what for us? We're going to talk about them in a couple of chapters from now. But what are white blood cells for? Disease. Keep us protected from foreign substances. So foreign substances can get in anywhere, right? So they need to hitch a ride throughout the circulatory system to get where they need to go. Red blood cells, what's their main job? Oxygen. Carrying not only oxygen, but carbon dioxide too. Okay? So respiratory gases are going to hitch a ride on erythrocytes. And then the other part of that less than 1% is something called a platelet. And a platelet is actually fragments of cells. Where are these cells born? They're born in the bone marrow. And how do they get into the circulatory system? Any ideas? They squeeze their way from the bone marrow into capillaries. And that's how they get into the circulatory system. We're going to talk about platelets and learn that the cell that creates the platelet gets so big. It's called a megakaryocyte.
When you say big.
Big, like size big. All these cells come from the same cell. It's called a hematopoietic stem cell. And what causes it to change into all the different cells is some sort of signal. Any ideas? Oh yes, you've learned one already. Actually, you learned two. Near the end of the chapter, on your list of things to know. There's a hormone in there. Begins with an E. You getting there? No? I'll give you a hint. Erythropoietin. When do you think I secrete, and who makes it? Kidneys. So why do you think I would secrete erythropoietin from the kidneys? Because I need more red blood cells. And that hormone is going to come in contact with its receptor on its target cell. And what do you think the target cell's name is? Nobody? The hematopoietic stem cell. When it comes in contact with that cell, what's it going to cause? That cell to become somebody different. Who's that cell going to become? It's going to become an erythrocyte. So it's going to change what it does. See how the hormone thing works? And it's going to cause the cell to proliferate, mature, and differentiate into somebody else. A eventually mature erythrocyte. Yes? Different hormone, different result. Same cell. So that cell, the hematopoietic stem cell, has a whole bunch of different receptors on it. Yes? Receptors for different chemicals that when that cell becomes stimulated by a different chemical, another hormone or some other chemical, it's going to cause it to do something what? Different. Change into a different cell. You following me? Yes?
Is it the same process for white blood cells?
Different chemicals. Same process. Okay? Same cell. So that's great. I got one cell type. It can be a whole bunch of different things. When we talk about stem cells, they have the ability, in the case of the hematopoietic stem cell, when they become stimulated by different signals, to proliferate, make more of themselves, and change into something else, differentiate. Not all our mature cells have the ability to do that. When we were first conceived, we come from one cell, correct? That one cell has the ability to do what? Make more of itself and be a whole bunch of different cells. But once those cells mature, typically, unless there's some sort of stem cell, they're who they are forever and ever. Yes? So that's the basis of what kind of research? Stem cell research. Where's the best place to get really good stem cells? From a developing ball of cells. Yes? Do you have some in your body that can do that? We met some when we talked about special senses. When we had cells, basal cells, that were able to differentiate and become new receptors on our tongue and in our nose. And these guys, the hematopoietic stem cell. So if I look at a tube of blood, I draw a tube of blood in the lab. And anybody not have a blood test in here? Everybody's had a blood test, right? They go and draw some blood. Well, what the heck are they doing? Well, they're going to take this whole blood, which is plasma and cells, and they can test your transport mechanisms to see if you're getting enough glucose, or you're getting enough sodium, or you're getting enough potassium, because this is where it's going to hitch a ride. So in the clinical lab, when I take a tube of blood, I can do tons of different tests to see if everything is within normal range in your body. There's thousands and thousands of different things I can test for that are traveling through the circulatory system. So many of the tests that you do in the lab is done on the liquid portion, plasma. And sometimes I can look at the numbers of cells that are hitching a ride in your circulatory system and see whether or not everything's running smoothly with you as well. So if you have the right number of red blood cells. What if you don't have enough red blood cells? What might be your problem? Oh my God, I'm so tired. I can't wake up. What might be wrong? Not enough red blood cells, not enough oxygen delivery, not enough energy. Yes? So a simple tube of blood can tell your doctor so much about what's going on with you. So the lab is an amazing place where wonderful things happen. Sorry, it's just coffee's kicking in. So we can look at the numbers of cells, we can look at substances within the plasma, and that little centrifuge, when I spin it around in there, will separate the different elements based on their weight. So the heavy stuff's going to do what? It's going to go to the bottom of the tube. The kind of in the middle stuff, and we're going to do this in lab, but we're going to do it on a small scale. We're going to take a little blood from our finger, and we're going to do a test to see how many packed red cells we have. That test is called hematocrit, packed red cell volume. So what would be a normal packed red cell volume if I looked at the numbers on this chart? Yeah, about 45%. So there's values in your textbook. You'll see a little bit different. Men tend to be a little bit higher. Women tend to be a little bit lower. Why do you think men higher, women lower? Yeah, men have tend to have a higher muscle mass. More muscles need more what? More oxygen. Okay? So if you look at, say, two females of the same size, if one works out and one doesn't, who might have a higher hematocrit? The one that works out. Yes? So, this can tell us a lot about what's going on with us. So the components of blood, physical characteristics, talk about where it's distributed throughout the whole body. It helps to regulate, it helps to protect. It's basically our delivery system, our maintenance. Blood plasma is not just water. So when I draw that tube of blood and I separate it out in the lab and I look at the plasma part, plasma is mostly water. And according to your textbook, it's about 90% water. But the other 10% is all of these things that are being transported. Now, some of them are things that are going to help maintain different functions in our body, and some of them are waste products that are being carried to places like the kidneys for disposal. So what am I going to find in plasma? Well, I'm going to find things dissolved in it. And solutes is the fancy chemistry way of saying things dissolved in water. Electrolytes, extremely important. What are electrolytes? Yes, so like sodium, potassium, chlorine, they're charged particles. We know how important those are because we had a big discussion about them last semester when we talked about the nervous system, when we talked about a muscle contraction. All of those were part of these charged particles that are flowing through blood plasma. And again, being delivered to cells. Plasma proteins, that's what makes it thicker than water. Those proteins are extremely important. A lot of them, and you see this word not only here under electrolytes, but you see it here under plasma proteins. Osmotic. What the heck is that? Osmotic pressure, osmotic balance. Something to do with water, yes? Remember when we talked last semester about osmosis and diffusion? What was the definition of osmosis? The movement of water from a high concentration to a low concentration. So these substances are also going to help maintain what we call fluid compartments in the body. When I talk fluid compartments, Chapter 27, Fluid and Electrolytes, at the end of the semester, we're going to talk about how important it is to keep just the right amount of water inside cell, outside cell, in between cells and tissues. So these substances in the right proportions are also going to help maintain water compartments throughout the body as well. Albumin, 60% of those plasma proteins are albumin. And you probably ate some this morning. Did anybody eat any albumin this morning? Eggs? Yes. The white of an egg? Albumin. So when I eat an egg, what's the most important part for me? The white part. You don't really need the yellow part. Although it's good when you have toast and stuff. Yeah, definitely. Making me hungry. So albumin, a major plasma protein that helps with osmotic balance. Has anybody ever seen those terrible commercials that always make me so sad when you see someone who's starving? Those beautiful little children that have those very large extended abdomens. Why is that happening? Yeah, they have such a huge protein deficiency that they have trouble keeping fluid in the compartments they belong in. So that fluid seeps out of their cells and into abdominal pelvic cavities. So that's just fluid that has leaked out of places where it should be in the circulatory system. So these are extremely important in balancing our fluid compartments. And albumin is a big player. Yeah, it's difficult and a long road, but getting the right nutrients into those children very slowly can help to even out fluid compartments. But that's what's happening when you see that state. Globulins, another one we're going to talk about in a couple of chapters. What's a globulin? I'll give you a hint. Typically we call them immunoglobulins. immunoglobulins. Immuno. Is that ringing a bell to anyone? Immune system. So these proteins, we're going to talk about 5 different ones again. 5 seems to be the number when we talk about blood. Remember MAG. You'll know what I mean when we talk about the immune system. But we have different immunoglobulins, protein in nature, called antibodies. And they are going to help neutralize foreign substances in our body and help protect us from foreign invaders. Alpha and beta are some different proteins that are produced by the liver, transport things, help with fat-soluble vitamin absorption. Those things are all important in many different reactions of metabolism. So plasma isn't just water. We have all this other stuff. Another group of proteins that we see usually floating around in the circulatory system in liquid form are a group of proteins called fibrinogen. We're going to talk about them near the end of this chapter when we talk about something called the clotting process. What's a clot? It's basically a plug. I get a hole in my circulatory system. What do I not want to do? Leak all over the place. How do I stop the leak? Plug it up. Plug it up with what? A plug. Some kind of plug, some kind of solid plug that will take normally floating around in my circulatory system soluble fibrinogen and turn it into a thread-like plug called fibrin. And the platelets are going to help me do this. Then we have a bunch of waste stuff, non-protein, nitrogenous, some things. It's kind of the stuff that makes your urine yellowish. Byproducts of breaking down old red blood cells and stuff like that. That's transported in the plasma to the kidneys so it can be sucked out and removed. Nutrients. Every time I eat something and I break it down in the digestive system, I'm going to absorb it into the circulatory system and get it to where it needs to go. So everybody ate breakfast this morning, right? Oh, gee, what am I going to do? I'm going to bring you breakfast. Goodness sake. So these things that you get when you eat breakfast or whenever you decide to eat are being transported by the circulatory system. Blood. Respiratory gas is a big one. So I breathe in. Then with the help of pressure differences, those red blood cells are going to have 4 nice little spots for my respiratory gases. Every hemoglobin molecule, which is, brush off the memory cells, a quaternary protein. What's quaternary mean? 4 little balls of protein units all kind of stuck together, and smack dab right in the middle of each of them is a little molecule called heme. And every heme can carry 1 respiratory gas. It's kind of like a amusement ride. Think of the Ferris wheel. Thank you. I'm going, trying to remember what that is. So there's 4 seats on every hemoglobin molecule. And when I pick up respiratory gases in the lungs, they're going to have a seat. And where am I going to dump? Where are they going to get off the ride? In the tissues. Who's going to jump on in the seat? Carbon dioxide. Yes? Then we go back to the lungs, and what's going to happen? Carbon dioxide's going to get off. Oxygen's going to get on. You getting the point? So that's what hemoglobin does. So respiratory gases, red blood cells are just basically big bags of hemoglobin that are going to help transport throughout. And then, of course, wonderful who? Hormones. So blood plasma isn't just a bunch of liquid. It's all of these things that are being transported through the body to help maintain homeostasis. You should know that chart on page 633. So the next thing we're going to talk about is what we call the formed elements, the stuff at the bottom of the tube. So 45-ish% is going to consist of formed elements. These formed elements come from the same cell. What was the name of that cell again? Hematopoietic stem cell, or hemocytoblast is another name for it. And that cell in the bone marrow, when stimulated, is going to become what we see hitching a ride in the circulatory system. Again, most of them are these guys. These guys are erythrocytes. They look funny, though. Something's missing. Who's missing? Nucleus is missing. Where the heck did the nucleus go? They don't have one when they mature. They had one in the bone marrow, but just before they come out into the circulatory system, it's kind of taken up too much space, so they spit it out. And then they go into the circulatory system. So erythrocytes have no nucleus. They're basically big bags of hemoglobin. Then we have different white blood cells. In this particular slide, all we see is 3 of the 5 that we could have. Those look different. They're what? They're bigger. And then what's this big purple hunkin' thing? The nucleus. We're going to find out the nucleus is very different in all the different types of white blood cells. And then I got some schmutz over there. What is that? That's the fragments of cells I talked about. Those are platelets. So when that cell that has matured from a hematopoietic stem cell goes to squeeze out into the circulatory system, guess what happens to it? It blows up. And the pieces are what's going to hang or hitch a ride in the circulatory system. Believe it or not, those pieces or fragments of cells are going to be extremely vital to forming that clot we were talking about earlier. So these are what we call the formed elements. The first one they talk about is the erythrocyte. Now, if I look at the erythrocyte under the microscope, it kind of looks like there's a little white hole there, right? Is it really a hole? No, it's just a really thin spot. And remember, my compound light microscope shines light right through it. So it's this part here. See the thin, skinny part? Why do we have the thin, skinny part? That's where the big honking nucleus used to be. When I spit it out, now I kind of have a thin spot. So this is sometimes referred to as a biconcave disc. Concave on both sides. And that biconcave disc is basically what? I spit out my nucleus, so what do I have left? Got a bunch of organelles, and then I have what? Hemoglobin. Big bag of hemoglobin. Now I'll hold on to my organelles, mitochondria especially, because I'm going to need a little energy during my life as a red blood cell. But basically I'm a place where you're going to find hemoglobin. So thousands and thousands of hemoglobin molecules are packed into an erythrocyte. So hemoglobin molecules are for carrying respiratory gases. So again, if I look at this hemoglobin molecule, I see these little things that look like frisbees. What are those? And what color is it? It's red. That's why they're red blood cells. Because this pigment, heme, which contains iron, is reddish. And each heme molecule can carry a respiratory gas. So there's a couple of terms in your book that you should know. When Hemoglobin is carrying oxygen. What do we call it? And this is found on page 635 in the second part of your book. It's called oxyhemoglobin. And when hemoglobin dumps off its oxygen, what do we call it? Deoxyhemoglobin. And when it's carrying carbon dioxide, what do we call it? Carbaminohemoglobin. So you should know those few terms that are referring to hemoglobin in different states. So depending on where I am in the circulatory system as a red blood cell, I might have different things sitting in those seats, right? If I just came from the lungs, I'm going to dump off my carbon dioxide and pick up oxygen. So we'll see this, a brighter red. If I'm down in the tissues, in the capillary beds, I'm going to dump off my oxygen and pick up what? Carbon dioxide. So now I get a little darker red. Okay? So blood that's making its way back to the lungs, and when we talk about circulatory system itself, where am I if I'm making my way back to the lungs? Actually, I'm making my way back to the heart so I can get a push to the lungs. I'm in veins. So blood in your veins is a little bit what? Darker than blood in your arteries, because blood in your arteries that have come away from the heart and now are being transported to your tissues is a little bit brighter red because it's carrying more what? It's carrying more oxygen. So, depending on where I am, I look a little bit different, and somebody different might be sitting in my seat. But again, I have 4 seats, and that's where what red blood cells are for. If you look at the top of page, and you're going to hate me for this, 636, we see a progression, a maturation process of these cells. And you're going to see them for all the cell types. Do you have to know them? Yes. I told you were going to hate me. So here's the stem cell. That's always the first one. So that's easy. That's going to be the first one for all of them. Because all of these formed elements come from that cell. Where is that cell found? What kind of bone marrow? Red bone marrow. So in the red bone marrow, we have the hematopoietic stem cell. When something comes in contact with this cell, it's going to cause it to go through this specific maturation process. What's that something? I heard there's hermits down for sale in the bookstore. Does everybody know that's my favorite cookie? Yes? So I must quickly run before the hermits get sold out and I need to get my share, correct? Running down the hall. Now the bookstore is quite far away, so I'm getting quite a workout. What happens? I start breathing heavier because I'm using up more oxygen. When oxygen levels go down, what do you think I might do? I need to carry more. How am I going to carry more? I need to make more, yes? So when oxygen levels start to go down a little bit, I'm going to do what? I'm going to breathe faster, but I need somebody to carry all this stuff. My heart's going to pump faster, yeah, but something else is going to happen. What am I going to do? I'm going to make more. How? Going to make more red blood cells. Erythropoiesis is the process, but how am I going to do that? Say it loud. Kidneys are going to beef up the release of right, erythropoietin. That's going to come in contact with that guy. That's going to cause more of these to come on board. Okay? So we're going to cause that cell to differentiate eventually into a mature erythrocyte. Just before I leave the bone marrow, and this is where I am, well, kind of here actually. I'm still in the bone marrow here. Excuse me. I'm going to get rid of my nucleus at this orthochromic erythroblast stage. And then I'm going to get spit out into the circulatory system. What's left here is some remnants of the DNA that I had in my nucleus. I can't see that under the microscope. I actually have to do a special test. What do you think might be going on if your patient has an increased amount of erythrocytes? Or they might, for some reason, need to make red blood cells at a faster rate than normal, right? Give me a reason why I would have to make more red blood cells at a faster rate than normal. I'm bleeding, I'm leaking somewhere, right? What if my red blood cells aren't as efficient as they should be? What if they don't have enough hemoglobin in them? What am I going to have to do? I'm going to have to make more. So those are some of the instances where we might see an increased number of these guys, the brand new red blood cells that kicked out, at a higher rate in our patients. This is the erythrocyte. Basically what happens is this kind of unravels and degrades. These look the same under the microscope unless you do that special test. Okay? So those are the guys that are in the circulatory system. Those are the ones we see. If we see this in the circulatory system, is that normal? No, that's way unnormal. Then something's going wrong. I shouldn't see nucleated red blood cells in my blood tube. Something's up. I might have a problem with abnormal maturation of those cells. When we talk about blood cancers, leukemias, those are abnormal progressions of some of these cell types. So we'll start to see, we won't see the normal version in the circulatory system. We'll see like earlier or abnormal ones in the circulatory system. So again, that tube of blood, we can tell so much about our patient. So some of the mechanisms that are going to regulate the production of red blood cells are described in your textbook, and we talk about hormonal controls and erythropoietin. So the process of that cell turning into a red blood cell is called erythropoiesis, and those are the different stages we see in the maturation process. So another reason why I might need to make more, which we mentioned before, is my red blood cells don't have the right stuff inside of them. So the breakfast skippers, the I don't eat when I'm supposed to eaters, guess what? If you don't get the building blocks you need to make your cells and your hemoglobin molecules the way you should, you're not going to make enough. You might be what? If you don't have enough hemoglobin in your cells, what might you be? You ever hear of someone who's anemic? One of the very important vitamins that are going to help the production of hemoglobin are the B vitamins and iron, right? Smack dab in the middle of my heme molecule. If I don't have the proper ingredients to make enough hemoglobin, I might not have proper oxygen carrying capacity. So they talk about some disorders on page 638 of your red blood cells. Anemias can be for many different reasons. We can be losing blood, what's called hemorrhagic anemia. Not enough red blood cells, pernicious anemia, problems with the substances I need to make the cells. For some reason, we might have destruction of my red blood cells. Certain drugs can do that, and they describe something called aplastic anemia. Certain diseases can do that as well. Yep. Too many red blood cells That's good, right? No, because what happens to my blood? Yeah, it becomes a little bit too thick, which is not good, because it can get what? It can plug up the works down in those smaller tubes. We're going to talk about the capillary beds. So too many red blood cells is not that good either. Thalassemias, different types of anemias that produce abnormally shaped hemoglobin molecules. So remember that ride with the 4 seats? Well, if you ever go to a really old carnival, some of the seats are what? Some of the seats are broken. So the ride isn't as efficient as it should be. So if I make the molecule the wrong shape, I can't get as many people on the ride. Sickle cell anemia, which is an abnormal one abnormal amino acid in that big, huge molecule that causes the hemoglobin molecule to be misshapen. So can I carry as many people on the ride? No. Oxygen carrying capacity is down. That's sickle cell anemia. Thalassemia, again, Mediterranean descent tends to have a higher incidence of this particular hemoglobin abnormality. Again, sickle cell anemia. Sickle cell. You know why? When you get malaria, do you know what it is? It's a nasty little parasite. It's called Plasmodium falciparum. And you know where it likes to hang out? Inside your red blood cells. So it decreases your oxygen carrying capacity. But it doesn't like sickle cells because they're the wrong shape and it's too crowded. So we see a higher incidence of patients with sickle cell anemia coming from places that have a high incidence of malaria. It's actually more beneficial to those people to have sickle cell in those high malarial incidence regions. Now, why do you think they survive? Why do you think sickle cell is even around? I mean, it's not a beneficial thing for us in general, but in those areas, it's going to allow them to do what? Survive longer and do what? Reproduce, pass that genetic abnormality to their offspring. Yes?
So is it really just an adaptation?
When we talk about genetic mutation, that's a tough discussion to have, but it's not necessarily, it's like survival of the fittest. So something changed. It didn't change because there was malaria in the area. It changed because it was a mistake. But the mistake happened to be what? It happened to be beneficial to the people in that area. If it's beneficial to them, it allows them to do what? Survive long enough to have offspring. They pass that genetic mutation to their offspring. That's what the whole concept of survival of the fittest is. So the mutation doesn't happen because of it, but it ends up being beneficial to the organism and passed on in groups where it's going to provide a benefit. Right? Does that make sense? Okay, this little diagram on page 637 talks about the mechanisms of action, little negative feedback mechanism for erythropoietin and the regulation of erythropoiesis. Again, oxygen levels are going to drive those changes. When oxygen levels go down, we're going to increase secretion of erythropoietin and consequently increase production of red blood cells. So what happens to red blood cells when they get old? Yeah, we gotta get rid of them. You know, they can only like tops 120 days. They're going to go out in circulation. under ideal circumstances. Sometimes I get rid of them at a faster rate depending on what's going on in my body. But we're good at recycling. Yes? And who's going to help me break down old red blood cells? A couple of different places. The spleen is one. The liver is another. So this diagram shows us some of the waste products or byproducts that we're going to recycle from broken down old red blood cells. In the liver, we have macrophages in connective tissue that are going to grab onto some of those old guys and break them apart. So we have things like bilirubin and iron, some amino acids, some of the portions of that hemoglobin molecule, the heme and the globin. These are all things that are kind of recycled, and waste products are secreted into the digestive, through bile, and who else picks up waste products and gets rid of them for us? The kidneys, renal system. And the waste products of broken down old red blood cells tend to be a little bit on the yellowish side. So that's why stool tends to be, well, it kind of gets brown, but has yellowish pigments in it. And what about your urine? That's where those yellow things come from in your urine. Some of the byproducts, waste products of broken down old red blood cells. So here's my sickle cell versus my normal cell. And again, in this patient, because of one little amino acid mistake, we're going to see that the hemoglobin molecule isn't big and fat and round, it's kind of collapsed. So it doesn't fill up the red blood cell or all the red blood cells nice. In the lab, you're going to look at a blood smear of a patient with sickle cell. And not all of them, no. And it depends on what state the patient is in. When the patient is in crisis, you'll see more of their cells that look like that rather than that. But not all of their cells are going to look like that. Okay? So what the cells look like under the microscope can tell us a lot about the patients too. If I look at the red blood cells under the microscope and they look like that big beautiful one and most of it is red and there's only a tiny little bit of white in the middle, nice hemoglobin content, right size, right shape, we're good. But if I look at the red blood cells and I see this big, huge, empty space in the middle, then I go, not enough hemoglobin. Or if the size of them is way too small or way too big, I can tell problems are going on with hemoglobin production in red blood cells.
What would, like, a person that had sickle cell, would they have, like, outward symptoms?
Yeah, joint achy, pain, tired. Fatigue is a big one. Okay, so that's my erythrocyte. Erythrocyte disorders, basically those anemias, you should have a basic definition. Polycythemia, too many, not so good. The Olympics are coming up, and maybe somebody out there is trying to cheat. Yes? You ever hear of blood doping? What do? Yeah, they give themselves more red blood cells. Good idea? Nope. How come? You increase your chances of forming what? Yeah, spontaneous clots that are going to plug up different places. Now, what if it just happens to be in your brain? That's not good for you. What if it happens to be in your lungs? Duh! Heart! Not good. So do you want to take that chance? Some of them want to win so bad that they do. But that's some of the consequences from doing something like this that could occur. They take some blood that they have packed away, they spin it down, they inject it. Yeah, it's their own blood. When you donate blood, what do they do? So they separate it. Did you know that? When you donate blood and somebody gets your blood, they're not getting the whole kit and caboodle. Yeah, they're only getting the red blood cells.
It's not like what
That can actually do the separation if you want to donate like plasma. Yep. So it takes off plasma, puts back extra water for you, so you have the right blood volume, and you can just take plasma out. And plasma, because it's got all of those things in it, can actually be very beneficial to a patient. So if I need, say, some certain chemical that you have in your plasma, and I don't need your red blood cells, why would you give me those? It's just going to cause the volume in my circulatory system to be off. So I can actually extract plasma and give that to patients. When I give patients blood because their oxygen carrying capacity is low, do they need all the fluid part? No, all they need is the red blood cells. So what you do when you go to donate blood, what they do at the Red Cross, is they put it in a bag. Now, you don't see what they do afterwards. What they do then is spin that bag down, suck off the red blood cells, wash them off really, really good, and put them in. Got to have some fluid so I can put them back in, right? Yeah, what kind of saline? Because we want to keep the cells shaped nice and happy. Isotonic saline, exactly. Normal saline, 0.9% saline. So that's what's in the bag that you see patients getting when they get blood. It's basically red blood cells. We don't have a good synthetic thing as of now to give patients that need that extra boost. So we still have to go and donate blood. We've tried in the lab for many, many years to replicate that, but we still haven't really done a really good job at it. So it's the good old blood donation is still the best way to boost oxygen carrying capacity in patients that need it. Well, because of all the other things that blood may carry, you know, and then for religious purposes, some people won't do that as well. Excuse me. Okay, so that's my red blood cells, erythrocytes. The next type of formed element they talk about in the book is white blood cells, or leukocytes. Now, here's the tricky one. There's 5 different kinds of leukocytes, and one of them is called a lymphocyte. Leukocyte, lymphocyte. That word's kind of close, yes? So don't get the two confused. A lymphocyte is a leukocyte, but a leukocyte is only one of the 5 is a lymphocyte. See what I mean? So white blood cells, that less than 1% in my formed element gamut, is of 5 different types. And generally, they're larger than red blood cells. They have a nucleus. They are drawn to things. So even though they take a hitch a ride in the circulatory system, that they're only hitching a ride there. They're eventually going to go someplace else, be delivered someplace else. Some of them are going to hang out in the circulatory system and act as surveillance in the circulatory system. But many of them are just hitching a ride. They're going someplace else. And what allows them to go there and get out of the circulatory system and get to where they're going? Some sort of chemical message is going to draw them to where they need to go. Remember last semester when we talked about skin and we talked about getting cut and tissue repair? Remember that? There was a weird little word in there that was that word meant drawing cells to the area chemically. No. Chemotaxis. Okay? So chemotaxis is that chemical message. It might be in the tissues. It might be in the bone. Hey, monocyte, come this way. You got to come and break me down. Yes? So they are drawn to other places via chemotaxis as a group. They might not necessarily stay in the circulatory system for long periods of time. Some of them are going to leave the circulatory system and go hang out in the liver and change into a macrophage. And what's a macrophage do for me in connective tissue? Yeah, it's going to phagocytize things. You see what I'm saying? So they don't necessarily stay in the circulatory system for long periods of time. They're basically hitching a ride to somewhere else. So we have 5 different types. Yes, your textbook finally has done a good job of visually representing the 5 different types. 3 of them, when I look at them under the microscope, they look like they have little bits of sand throughout their cytoplasm. We call these guys the granulocytes. The other 2 cells don't have as many of them. They are called agranulocytes. Cytes means what? Cells. 5. Remember. Ready? Never let monkeys... Wait a minute. You guys have your clickers? Okay. Everybody press your clicker. Remember, never let monkeys eat bananas. Yes? Remember that sentence because it's actually in order of highest to lowest in the circulatory system. So neutrophils, lymphocytes, see the percentages? Monocytes, eosinophils, basophils. Never let monkeys eat bananas. Those are the 5 different types of leukocytes in the circulatory system. We good? Everybody click? Click anything. Everybody? Who doesn't have their clicker today? Everybody has it?
Just click anything.
Just click anything. Good. We're good? So there's 23 of us here today?
This one?
Huh?
This one up.
Oh. All right, I'll do it again after. 24? Count. You count. Make sure. And don't forget these 2. So never let monkeys eat bananas. Did you write that down? Okay. So the granulocytes, and again, when I look, do we have? Okay. The only way I can tell the difference between one white blood cell and another is by their chemical components. So when we look at these cells under the microscope, what do we have to do to them? We can't just put a drop of blood and smear it out and look at them under the microscope and tell the difference between them, because they're all the same color. You have to do something called differential staining. The stain will pick up different chemical components in the cells. And the cells are named for the different parts of this stain that they bring out based on their chemical components. So the granulocytes, see them? They look like they have sand, but the sand is all different depending on the cell type. These granulocytes, the most numerous is this guy. He's called a neutrophil. The reason he's called a neutrophil is because the granules, those little pieces of sandy-looking stuff in the background, pick up the neutral portion of the stain combo. It's kind of pinky purple. That's how I describe it. And the granules are very fine, like sand. The other weird thing about these granulocytes is their nucleus. Now, the normal nucleus we look at under the microscope tends to be this nice little round thing, right? Not in these guys. In these guys, the nucleus gets all kind of weird shaped. It can be all sorts of weird shapes. Another name for these cells is a poly or a polymorphonuclear leukocyte, PMN. What the heck does that big word mean? Exactly. Poly means many. Morpho refers to the morphology. Nuclear, nucleus. So a whole bunch of weird shaped nuclei is basically what that word means. Again, if I didn't have the stains to pick up the different chemical compositions of the different cells, I wouldn't be able to tell the difference between them. So neutrophils are largest in number. They are phagocytic cells. So what do they do for a living? They go phagocytize things. When we talk about an immune response in a couple of more chapters, we're going to talk about this guy. This guy is going to get rid of the trash for us. If we have an antibody that neutralizes an antigen, it creates a little antigen-antibody complex. How do we get rid of it? That guy phagocytizes it, gets rid of it for us. We talked about this guy when we talked about wound healing. So we see some neutrophils that will migrate out of the circulatory system and go into tissue if I get a cut and help to phagocytize who? Any nasty little bacteria that might get in. Remember what phagocytosis is? What is it? It's cell what? Cell eating. We talked about that way back when, right? So that's what neutrophils are for me. The next one they talk about is something called an eosinophil. Still under the granulocyte, still that polymorphonuclear structure, but those granules are no longer little neutral pieces of sand. They're balls. And they're orange, reddish orange, because they pick up what portion of the stain? Something called eosin. So eosinophils are named for that reddish granule. Again, the granules are no longer sand, they're balls. Can you see that? It's better when you actually look at them under the microscope. But eosinophils are much less in number than the neutrophils. We're going to find those during things like allergic reactions or parasitic infections, because the granules in the eosinophil are going to help to neutralize different foreign substances. Even less in number in the granulocytes is this guy. This guy is called a basophil. Those little granules pick up the basophilic portion of the stain and turn the little balls what? They look black, don't they? Really, really, really dark, dark blue black.
Yeah, you can almost like mask it.
Yeah, because the granules kind of mask it because of the stain. So the nucleus is here, see it? But those black granules fill that. That's the smallest in number that we'll see floating around in the circulatory system. And again, the basophil contains chemicals that act to create inflammatory response. Remember when we talked way back when, connective tissue, we talked about mast cells? That's where they come from. So mast cells that release things like histamines that create what? Histamines create what? Inflammation, increased fluid buildup in certain areas. They hitch a ride in the circulatory system before they go to connective tissues, and they are the basophil. Yes? Everybody following me? So those are the three granulocytes: neutrophils, eosinophils, basophils. You in the lab are going to do something called a white blood cell differential. So guess what you're going to do? We're going to make a blood smear, and we're going to look at the cells, and we're going to count the white blood cells. And guess how many we're going to count? 100. And we're going to keep track of the 100 we count. Guess what's going to be the most? Neutrophils. Guess what's going to be next? Lymphocytes. Never let monkeys eat bananas, yes? So the agranulocytes are these two guys, the lymphocyte and the monocyte. Lymphocytes are different because they're going to have a nice big round nucleus. Now some of the lymphocytes have a nucleus that are going to fill almost the whole cell with very little cytoplasm. That's this guy. He's called a small lymphocyte. But we also have larger lymphocytes. The characteristic for this guy is the nucleus, which is going to be very densely packed and round. And then the cytoplasm. The cytoplasm is very different color from something like this, isn't it? It's actually very blue. In the lab, when we learned how to differentiate these under the microscope, they were described as robin's egg blue. So the cytoplasm of a lymphocyte takes on a lot of blue from the stain combination that we use. What's a lymphocyte for? We're going to talk about these guys when we talk immunity. These guys are going to help me protect against foreign substances by producing chemicals, some of them, called antibodies. Those are my friends, the B cells. And then other ones, called T cells, are going to help organize everybody in an immune response. I call the T cells the cruise director of the immune response. Because without the T cell, nobody knows what to do. So when something foreign comes into my body, I have cells that will recognize it as foreign and kill it. But if it makes it past those guys, these are the guys that are going to kick in. They will be able to recognize them very specifically as foreign and wage a very specific attack against them.
That's why AIDS patients, it's so important to track their T cells.
Correct. And why is it a problem with AIDS patients? The nasty AIDS virus hangs out in T cells because a virus actually takes over your cellular replication mechanisms to make itself. That's how it survives. And the only way for it to get into your cells is to have a very specific, I call it the door in. And it's one of the proteins. It's called CD4. There's a whole bunch of proteins on the surface of cells. But the viral door in is called CD4. And guess where I find CD4? On T cells. So that's how the virus gets into the T cell. Now, remember, that was the cruise director of an immune response, right? If that cell can't do its job and organize everybody in an immune response, everybody's like, ugh. Yes? So that's the problem. When we get to the point where that virus is really wreaking havoc in a patient, they develop something called AIDS, acquired immune deficiency syndrome. Correct. So you could be HIV positive and still have fairly normal immune function. But when the virus really wreaks havoc, then you cannot wage a war against foreign substances. So patients succumb to things that, are normal things in our environment that we are fighting off all the time. But they can't do that because they don't have a proper response to those antigens, foreign substances. We'll talk about that when we talk immune system. So that's my lymphocyte. B cells, T cells. B cells make antibodies. Antibodies are chemicals that are going to help neutralize foreign substances. T cells, cruise director. They're going to help do a whole bunch of different things in an immune response and make sure the whole thing works well. And then the last one, just kind of hooking a ride in the circulatory system, is the what? Monocyte. The monocyte, big cell, that's the biggest of the white blood cells. Very different looking. The nucleus can be round like that, but sometimes it looks like, and most of the time, it looks like a big kidney bean. It's not tightly packed like the lymphocyte. It's much more, I always describe it as airy. So you can see white coming through it. Same thing with the cytoplasm. It tends to be bigger and kind of fluffy, not nice and tight like the lymphocyte. It tends to be on the grayish side. So the monocyte has that bigger size, that kidney bean shaped maybe nucleus, and a grayish cytoplasm instead of a bluish cytoplasm. And we've already met the monocyte. What's it do for a living? It can. What's it do mostly, though? Yeah, it tends to phagocytize things, break things down, tends to like leave the circulatory system. It's just kind of hanging out there. Yes? We will find it associated with some sometimes an immune response. Most of that's the job of the lymphocyte. Okay, here's another progression for you. Hoy. The red blood cell was easy, right? Remember, I come from one cell. All formed elements come from one cell. Well, how does that one cell change into 5 different things? There's going to be 5 different things that'll cause me to change, right? Now, there's a chemical name for all of them. I want you to remember 2 words. Interleukins and cytokines. And these are basically groups of different chemicals that will cause that stem cell to become one of the 5 players with respect to leukocytes. And I'm not going to make you memorize the specific ones. Interleukins and cytokines, groups of chemicals that are going to cause those white cells to be produced. Yes? Okay, so if I get stimulation to become an eosinophil, this is my maturation. If I get told to be a basophil, that's my maturation. Neutrophil, that's my maturation. Are you going to have to know that? Yes. But notice something. Same, right? Then we change and we get the name of the cell. Yes? So we get eosinophilic myelocyte, basophilic myelocyte. And it's the same, okay? So it's not bad. We're going to be a myelocyte, then we're going to be a band. If I look under the microscope at my white blood cells and I see lots of these, what might that mean? Huh? How do you know I'm sick? Because I'm doing what at a faster rate? Yeah, I'm kicking them out faster, and I have a higher percentage of what we call the band. So if we look at the results of that white blood cell differential, and the technician notices that there's quite a few of those cells that look like that, and not that really weird shape, they're going to note that. Because that might tell the doctor, something's going on. This patient's making white blood cells at a fast rate. And they can look into that.
Can you spell the same?
I'm trying to, where is it in the book? Production and lifespan of leukocytes. Colony-stimulating factors, interleukins. It's on page 642 under production and lifespan of leukocytes. Yeah, they didn't write cytokines in there. Cyto, C-Y-T-O-K-I-N-E-S. Interleukins and colony-stimulating factors. And that's what a cytokine is. Okay? So, also different numbers of these guys. If they don't fall into the right percentage, I'm making too many neutrophils. That might mean I have something I need to phagocytize that's hanging out in my body. Or I'm making too many lymphocytes. Maybe that means I have an immune response going on, and I'm making a lot more antibodies than normal. This down here I want to point out. These are all in where? These are in the circulatory system. Where are these? In the bone marrow. Am I supposed to see any of these in my circulatory? Nah. So if I start to see these in my circulatory system, there's a problem. Okay? In tissues, these guys that are hitching a ride can actually squeeze out of the circulatory system, and that's what I see down here on the chart. So monocytes, when they squeeze out of the circulatory system and go into the tissues, what do I call them? Macrophages. B cells, B lymphocytes, when they squeeze out of the circulatory system and go into tissues, they become plasma cells. And these are the guys that make what? These are the guys that make antibodies. T cells can also squeeze out of the circulatory system, and they become something called effector T cell. And that's the guy that's going to help me do what? Organize that immune response. Okay? So this little part of the diagram is in the tissues. And see how it says some become, some become, some become. Depending on what's going on, they'll squeeze out and do their thing. Now there's a couple of confusing, and you'll notice this if anybody picks up on some of these little nuances in our books. There's a whole bunch of different what we call normal values. They're different in your lab book than they are here. There's actually two different groups in this one chapter. So I don't want you to memorize numbers, but I want you to get a sense of what I would find in a drop of blood. I'm going to find millions of red blood cells in one drop of blood. That one drop is called a cubic centimeter. Okay? And if I take a centimeter, cubic millimeter, if I took a cubic millimeter and made a little box, how much blood could I fit in it? One drop. Yes? So think, one drop. There's about 4 to 6 million erythrocytes, millions, in one drop of blood. There's thousands of white blood cells, 4.8 to 10.8 thousand per drop. The numbers I do want you to know are on that diagram on page 641. So I want you, as far as normal values for leukocytes, I want you to know these. So neutrophils, 50 to 70% of my neutrophils are going to be, 50 or 70% of my white blood cells are going to be neutrophils. Then monocytes, 25 to 45%, lymphocytes, 25 to 45. Monocytes, 3 to 8%. Then we have eosinophils, 2 to 4, and basophils, 0.5 to 1. Okay? And if you remember what? Never let monkeys eat bananas. You're going to memorize them in order. Did everybody push the button when I said push the button? You sure you want me to put it on again? Buttons have been pushed. All right, good. All right, we'll pick up with leukocyte disorders on Thursday. Now, guys, just a heads up. Guess what happens after we finish this chapter? Tests. So I am going to do you a little favor and maybe throw some hints up there for you. Maybe I'll do that today. Give you a little heads up. Okay?